So have you ever stopped and wondered exactly where plants get their food? I mean, it's a beautifully simple question, right? But the answer actually holds the key to literally the greatest energy puzzle on Earth. Every breath you take, every move you make, and every single bite of food you eat connects right back to this microscopic machine operating silently in the green leaves all around us. Today, we're going to solve that puzzle.
Welcome to The Explainer. I am so thrilled to have you here as we dive into a really fascinating macro to micro journey into nature's ultimate energy factory. We're talking about photosynthesis in higher plants. We're going to zoom all the way down to the molecular level to figure out exactly how sunlight gets transformed into the chemical energy that powers our entire world.
Okay, to make sure we stay on track, here's our roadmap. First up, the plant food mystery. Then we'll head inside the chloroplast factory, check out the color of photosynthesis, watch the Z-scheme in action, and wrap it all up with powering up ATP synthesis.
All right, Section 1, the plant food mystery. Autotrophs and heterotrophs. You know, it's a fundamental truth that all animals, and yeah, that includes you and me, rely entirely on green plants, our autotrophs, to synthesize our food. Basically, we're just heterotrophs living in an autotrophs world.
Now, it actually took centuries of scientific detective work to figure out how they pull this off. I mean, we went all the way from putting mice in glass jars to understanding really deep molecular chemistry. Back in 1770, Joseph Priestley figured out that plants could somehow restore the air that breathing animals damaged. A few years later, in 1779, Jan Ingenhousz proved that sunlight is absolutely essential for this whole purifying process. Fast forward a bit to 1854, and Julius von Sachs discovered that plants are actually producing glucose as they grow. And then, a massive, mind-bending breakthrough hit in the 1930s when Cornelius von Neal demonstrated that the oxygen plants release, it actually comes from water, not from carbon dioxide.
The culmination of all that historical research gives us the absolute master equation of photosynthesis. It is so brilliant and perfectly balanced. Exactly 6 molecules of carbon dioxide and 12 molecules of water go in. You add some light energy, and what comes out? One complex molecule of glucose, which is the exact sugar powering the global food chain, along with 6 molecules of water and 6 molecules of oxygen. I really want to emphasize this breakthrough from Van Neal because it's just incredible. By studying purple and green bacteria, he proved beyond a shadow of a doubt that photosynthesis is a light-dependent reaction where hydrogen from water reduces carbon dioxide. Just think about that for a second. That means the specific oxygen molecules you are pulling into your lungs right now were quite literally born from the splitting of liquid water. No way, right? Moving right along to Section 2, inside the chloroplast factory. We are now zooming way past the surface of the leaf, deep inside its inner cells, entering a strictly microscopic domain.
Welcome to the chloroplast. I want you to think of this organelle like a highly organized, microscopic factory, and it has a very strict division of labor. It's basically got 2 main environments. First, there's a complex membranous system designed purely for trapping incoming sunlight. Then, there's a fluid-filled matrix called the stroma, which is where the enzymatic synthesis of sugar actually goes down.
Let's contrast those 2 main factory floors for a second. The light reactions versus the dark reactions. The light reactions happen right in those membrane structures. They're directly driven by light, and their whole job is to pump out these high-energy biological batteries, ATP and NADPH. The dark reactions happen out in the fluid stroma. Now, a quick heads up here. Dark reactions is honestly just a naming convention. It absolutely does not mean they happen in the dark. They're just light independent, meaning they rely on the ATP and NADPH produced by the light reactions to actually synthesize the sugar.
All right, Section 3, the color of photosynthesis. So now that we know where the factory is, let's look at the vibrant solar panels of the plant that actually trap all that incoming light.
When you glance at a leaf, it probably just looks like one flat shade of green. But scientists use a process called chromatography to reveal that it isn't just one color at all. It's actually a beautiful mosaic of 4 different pigments working perfectly together. We've got chlorophyll A, shining bright or blue-green. We've got chlorophyll B, which is more of a yellow-green. Then there are xanthophylls in yellow, and carotenoids ranging from yellow to yellow-orange.
So the really crucial takeaway here is that chlorophyll A is the chief pigment. It's the primary engine of this whole thing. The other 3, we call those accessory pigments, and they serve 2 vital functions. First, they act like a supportive net, absorbing a much wider range of incoming light wavelengths that chlorophyll A might just miss, and then they hand that energy over.
Second, they act as an essential protective shield, keeping chlorophyll A from being completely destroyed by dangerous photooxidation when the sunlight gets super intense. Which brings us to Section 4, the Z scheme and action. This is the dynamic climax of our molecular machinery where things get truly energetic.
So what we have here are essentially the two main pinball flippers of the plant's light harvesting complex. Photosystem 2 has a reaction center called P680, mainly because it absorbs red light perfectly at exactly 680 nanometers. Photosystem I has a reaction center called P700, absorbing at 700 nanometers. By the way, a fun little quirk here. They're actually named in the order they were discovered historically, not the order they function. So as we're going to see, Photosystem 2 is actually the one kicking off the whole shebang.
Let's narrate this Z scheme in action, because it's all about constant movement. Step 1. Photosystem 2 absorbs that red light, and the energy causes its electrons to become insanely excited. I mean, they literally jump into a higher orbit. Step 2. These excited electrons are scooped up and power their way downhill through an electron transport chain. Step 3. The electrons reach Photosystem 1, where they get blasted by more light and are excited all over again. And finally, Step 4. These re-energized electrons move downhill one last time to charge up NADP+ into NADPH. Boom, we are successfully charging the factory's batteries. But hold on a second. If Photosystem 2 just keeps firing off its electrons like that, how does it not just run out? Well, that brings us to this incredibly essential equation, the splitting of water. Two water molecules are quite literally ripped apart to create 4 protons, 4 electrons, and 1 oxygen molecule. And I am going to relentlessly remind you of the scale here. This reaction, happening right now on the inside of the thylakoid membrane, is providing the exact electrons needed to keep this machine running. And that oxygen molecule, that is the literal source of the oxygen you are breathing to stay alive today. Just amazing.
And that leads us nicely into Section 5, powering up ATP synthesis. We've seen a ton of chaotic electron bouncing, right? But how does the plant package all that chaos into usable cellular energy?
To explain this, we use the highly complex chemiosmotic hypothesis. But don't worry, we can break it down into 4 totally logical steps. First, that water splitting we just talked about leaves a bunch of protons trapped inside the thylakoid's inner space, which we call the lumen. Second, as those electrons transport across the membrane, they physically pump even more protons into the lumen. Third, on the outside, in the stroma, an enzyme is actually pulling protons away to make NADPH. The result? We build up this massive, highly pressurized gradient of protons inside the membrane. And finally, step 4, this intense pressure gradient eventually just breaks down, and the protons come rushing out to synthesize ATP.
And what exactly do they rush through? An absolute marvel of biology called ATP synthase. This is an enzyme featuring a transmembrane channel called CF0 and a protruding head called CF1. I really want you to picture this enzyme as a microscopic spinning turbine. As the trapped protons aggressively diffuse through that channel, the sheer physical energy of their movement forces the CF1 head to undergo a conformational change. It literally clicks together ADP and inorganic phosphate to synthesize ATP. The ultimate energy battery has officially been created.
Okay, so what happens next? Well, those freshly minted ATP and NADPH batteries head straight over to the dark reactions to start building sugar. Enter Melvin Calvin, the brilliant mind who earned the 1961 Nobel Prize for figuring out exactly how that happens. Right after World War II, seeing the absolute horrors of atomic weapons, Calvin and his team decided, you know what? Let's put radioactivity to a beneficial use. Using a radioactive isotope, carbon-14, he meticulously mapped out the entire pathway of carbon assimilation. He showed the world exactly how the energy captured by the light reactions is used to physically construct the sugars that sustain literally all life on Earth.
So, as we wrap up this journey from the broad green leaf all the way down to the spinning turbines of the thylakoid membrane, I want to leave you with one final, kind of provocative thought. The foundational principles of photosynthesis, established by Melvin Calvin, are right now being intensely studied by scientists all over the globe. If we can deeply, truly understand this flawless molecular machinery, could we soon use these exact principles to create our own completely clean, renewable solar energy? I mean, nature has already spent billions of years perfecting the ultimate green technology. We just have to learn how to mimic it. Thank you so much for exploring the brilliance of biology with me today on The Explainer. Keep learning, keep questioning, and I'll see you on the next one.